Good afternoon, everyone. Welcome to the second episode in our Bench to Bedside webinar series titled Harnessing Omics. This webinar is being recorded and will be available on our YouTube channel next week. We will be taking questions at the end, but please do submit your questions throughout the webinar. Joining us to present today's webinar is Tariq Mughal, Professor and Consultant in Hematology and Medical Oncology. Tariq has trained in London and Denver and practices in both Boston and London. He has been a pioneer in precision medicine efforts for patients with hematological malignancies and particularly chronic myeloid leukemia. Tariq is also the founder of Alpine Oncology Foundation, a Swiss-based cancer charity which focuses on providing access to molecularly targeted and immunotherapies for suitable patients in economically disadvantaged countries. I will now hand over to Tariq, who we are delighted is able to join us live from Boston today.
Well, thank you very much. It's morning here, but good afternoon to all of you in the UK. It's a privilege to be invited to give this particular presentation. It's fairly close to my heart, and I think many of you would be familiar that the term precision medicine, often used interchangeably with personalized medicine, has been introduced in cancer medicine recently to reflect the advances in our ability to characterize cancers at a molecular level and develop targeted treatment that might help counteract them. Now, you could say, arguably, the principles of precision medicine has actually been a cornerstone of medical practice since the earliest efforts to classify disease and prescribe specific treatment based on diagnosis. What is new, however, is the astonishing pace of advances in genetics and the impressive diagnostic, in particular, omics and treatment options. And that will be my principal focus. Okay. So this is my opening slide, and I wanted to emphasize how precision medicine actually impacts all aspects of cancer medicine from. Can I move to the next slide, please?
Hi, Tariq, you should have control on your end as well.
I do, but it's not moving. Yeah, that's it. Yeah, thank you very much. Yeah. So I was saying precision medicine impacts many aspects in cancer medicine, prevention, diagnosis, risk stratification to therapy. And according to the paradigm of precision medicine, the administration of agents targeting the molecular alteration is actually meant to reduce uncertainty in the clinical management. But sadly, it can also lead, paradoxically, to increased levels of uncertainty. And this is why I elected to use the title, The Paradox of Precision Medicine. And I will discuss some of these aspects. I think these slides are not moving from my side. Yeah, you can do that. Would you mind driving the slides when I say next?
Yeah, okay. No problem. We're here for you, Tariq.
Yeah, thank you. I'm sorry why it's not moving from my computer. So these are my disclosures. And next slide. And what I'm going to aim is to divide my talk into 4 sections. I will firstly talk about some historical milestones. And secondly, I will talk about approaches to molecular assays as part of the omics. And thirdly, clinical outcomes or clinical utility of these tools. And finally, we'll finish off with some concluding thoughts. Next slide, please. And historically, the lessons garnered from our patients with chronic myeloid leukemia, which of course is a rare leukemia, appear to be seminal and moving us closer to the prospect of precision cancer medicine. The CML success story has unfolded over a relatively short period of time, and the efforts of Janet Rowley, shown on my left, and John Goldman on my right, who was my long-term mentor and friend, have been crucial to our understanding of the biology of what is now considered a genetically simple disease. John and Janet's work has provided vital insights over the last three and a half decades that have resulted in the success of molecularly targeted therapy, not only for CML patients, but also for other malignancies, in particular common cancers like lung cancer, breast cancer, colorectal cancer. And these efforts have, of course, helped garner a qualified enthusiasm for precision medicine. Sadly, we lost both John and Janet within a week of each other in December 2013. Next slide, please. So as a consequence of this work by John and Janet, the tyrosine kinase inhibitor, imatinib, was introduced in the clinics in 1998 to target the only driver mutation in chronic myeloid leukemia, which is the BCR-ABL1. And the results were astonishingly good, and the drug was approved in 2001 after one of the shortest drug review processes on record at the FDA. It was less than 10 weeks. And then some 2 decades later, there was a long-standing, a long-term follow-up and review published in the New England Journal of Medicine, which confirmed the utility of this particular drug and how it was according most, if not all, patients to have a general survival, overall survival, which was similar to that of the general population. So you could almost say that the majority of these patients were at least operationally cured from chronic myeloid leukemia after being treated with imatinib. Next slide, please. And so today, we have numerous targeted therapies, both molecularly targeted therapies and, of course, more recently, immuno-oncology therapies, which have been licensed worldwide. And as illustration on this slide, I am just showing you a handful of these drugs which were matched with the key driver mutations, which are also shown in this slide in diverse cancers, beginning with HER2 testing and trastuzumab for breast cancer, EGFR mutation, then alotinib, and then more recently the BRAF mutation testing and drugs for melanoma, and so forth. And it's also important to note that particularly in the last 2 years, since 2017, the FDA has approved not only 20 targeted and immunotherapies, but also the landmark licensing of 2 chimeric antigen or CAR T-cell therapies, and also the first 2 histologically site-agnostic biomarker-defined immunotherapy. Now, the approval of a histology agonist therapy is remarkable, and that signals an important shift in the drug approval process because it breaks from the historical tradition of making decisions on treatment based on tissue of origin. And this is something still which a lot of us are absorbing and cannot believe it's already taken place. And the jury is still not in. But the 2 drugs, the first is pembrolizumab, which is a PD-1 inhibitor or immune checkpoint, which was licensed for the treatment of adults and pediatric patients with unresectable or metastatic solid tumors, which expressed 2 particular biomarkers. which is known as microsatellite instability high or mismatch repair deficient tumors. And then, of course, some 7 months ago, in November 2018, the FDA granted approval for the first NTRK inhibitor, which is known as larotrectinib, and this targets NTRK fusions for a variety of solid tumors, again, without recourse to histological proof. So these have been truly remarkable. And of course, at the bottom of the slide, I also acknowledge the Nobel Prize, which was accorded to two people, one in Houston, Texas, and the other in Japan, for their work on the immunotherapies, in particular the immune checkpoint inhibitors. Next slide, please. So having said that, we should now focus on the tools, the omic tools, in particular the molecular assays, which you need in order to make this precision medicine a reality. So let's see where we stand with that. Next slide, please. And here, of course, it's important to acknowledge that the discovery of DNA, following much deliberation in this particular pub shown on this slide, the Eagle pub, which many of you might be familiar with, in 1953, ultimately led to the availability of diverse genetic tests. And soon thereafter, a lot of pioneering efforts took place, particularly at the Sanger Institute in Cambridge, which led to the availability of the first generation PCRs, or polymerase chain reacting methodology, which was initially DNA and then subsequently RNA, and of course the first generation genomic sequencing, which is known as the Sanger sequencing, which became available. With these availability, the notion of cancer genomic complexity was recognized. And next slide, please. Next slide, please. And when trying to examine the genetic complexity through the genetic test, I often remember this particular ancient Indian fable shown here on this slide of the blind men and an elephant. The fable describes a group of blind men coming to different conclusions about how an elephant looks like by feeling different parts of the animal. The parable, of course, implies that one's subjective experience may be true, as shown here, but is inherently limited by its failure to account for the alternate possibilities or the sum total of facts. Next slide, please. So let us now examine the types of alteration which you get in cancer gene, because these are the ones which will be examined by the genetic test. Okay, so the first on the left is, of course, the normal cancer gene, and on the right is a copy number alteration. And here I show you the substitution, which is shown in yellow in the P-arm. And then here I show you the insertion and deletions shown here. And the next slide. And lastly, I show you an example of rearrangement of cancer gene. And this, of course, it was epitomized in patients with chronic myeloid leukemia, where you have a rearrangement between chromosome 9 and chromosome 22, resulting in the 922 translocation, or the Philadelphia chromosome, which harbors the BCR-ABL transcript. Next slide, please. And it's pretty clear that the each class of the genomic alteration present unique challenges for highly accurate detection by the current sequencing methodologies, which is known as next generation sequencing or NGS. And for example, if you look in the middle of the slide where we talk about the genomic rearrangements, it's quite clear that the breakpoints are often in the interim, which involves a very large search space. For example, in the case of CML, BCR-ABL, this is as large as 8.6 kilobases. And then, of course, when you have the rearrangement, you can have multiple possible partners, which adds to the complexity. And also, when you're looking at copy number alterations, it's important to identify and detect focal events. And if, for example, in the sample you have collected from the patient, the tumor purity is low, then the signal to noise phenomena is very diluted, and one may get errors introduced into the test. And also, very commonly, especially in solid tumors, you can have a problem of aneuploidy, and this can impact on the copy number alterations, and one needs to develop the appropriate mathematical algorithm in order to correct for the absolute copy number. So these are just some of the challenges which I wanted to share with you in a rather simplistic manner, which affects how the tests are conducted and how the results may be reported. So next slide, please. And here I summarize for you the currently available next generation sequencing approaches. Of course, firstly, you have the whole genome sequence, which determines the complete DNA sequence of the cancer genome, which remains very expensive, but more importantly, it's very time consuming, and it's only utilized in the research setting. The second is the whole exome sequencing, where you selectively sequence only the coding areas, which are the exome of the genome. And this is very popular, and this is the technology which has been utilized by a majority of the academic centers in the UK, in the US, and elsewhere. And then thirdly, we have the comprehensive genomic profiling, which is a massively parallel sequencing of the entire coding region in a defined subset of genes of interest. And this detects all of the 4 classics of alteration, which I showed you in the earlier slide. And this is a test which has been marketed by a number of companies on both sides of the Atlantic. And over the last 5 or 6 years, I have been involved with a company called Foundation Medicine on the medical side, and they have developed such tools. But there are other companies in other parts of the world which have also developed similar tools. And lastly, we have what is known as of genes of interest. And this perhaps is the most widely utilized genetic test. It's relatively cheap and reproducible, and doesn't require a lot of intense labor and bioinformatics, but it would only detect what you were actually looking for, in contrast to the previous test, which is the comprehensive genomic profiling, where you're looking at a range of different genes, and you were hoping to find what you're looking for, but you often end up getting a lot of other information which may or may not be relevant immediately or subsequently in the management of the patient. Next slide, please. Now, before you commence to utilize any of these genetic tests in the clinics, whether it's in clinical trials or in the real world scenario, it is important to establish that these tests have been validated appropriately, both analytically and clinically. And then, of course, the payers in particular, for reimbursement purposes, will require a robust evidence of the clinical utility of that test. Now, for the sake of people in the audience who may not be familiar with this terminology of the different validations, I have defined it as analytical validation, referring to the capacity to make accurate and reliable measurement of the candidate biomarker. And the clinical validity refers to the ability of the genomic test to accurately and reliably identify or predict a relevant endpoint. Next slide, please. So as illustration, I will show you a couple of the Foundation Medicine sequencing tests. And here I show you the result of the analytical validation of the first test, which is known as the FoundationOne test, which involved a panel of about 300 cancer-related genes. And the validation confirmed the utility of the test, both for mutation detection and copy number alteration in about 3,000 to 4,000 patients with diverse advanced solid tumors. And these results were published in 2013 in the journal Nature Biotechnology. And the test was readily available commercially, not only in the US, but in throughout the world. Next slide, please. And the second test in which I personally was involved in the validation, both the analytical and clinical, is the FoundationOne Heme test, which in contrast to the previous test, is sequencing both DNA and RNA. And here you sequence just over 400 entire coding regions of the cancer genes at the DNA level and about 265 at the RNA level. And this test is optimized for all hematological malignancies, sarcomas, and pediatric solid tumors. Because this test incorporates the RNA sequencing as well, which is more important when you're determining the presence or absence of fusion genes. This is particularly attractive in those cancers where fusion genes are playing a pathogenetic role in the cancer. Next slide, please. Next slide. And then more recently, over the last 7 or 8 years, the cell-free DNA, also known as the circulating tumor DNA or liquid biopsy, has become available. And this was developed by a number of academic centers. And here in the UK, a lot of work was done by a colleague of mine, and we'll pay tribute to him momentarily. But I should emphasize that this test is, at the present time, is particularly positioned for monitoring the progress of a patient with cancer who's being treated with therapy, because it's relatively easy to do the blood test. And work done at University College Hospital, led by Charlie Swanton, has very nicely demonstrated the use of this liquid biopsy to monitor patients with non-small cell lung cancer being treated with targeted therapy. And in the hematological landscape, is particularly useful for monitoring MRD or minimal residual disease presence. Next slide. And I think here we must pay some tribute to the late David Grimwade, who was a close friend of mine and a professor of hematology at King's College and Guy's Hospital, who did a lot of pioneering work on the use of ctDNA, largely in hematological malignancies and particularly in acute promyelocytic leukemias. Next slide, please. I will now switch to another test which has become very popular in the last 3 or 4 years, particularly in regard to selecting patients who may or may not benefit from the use of immuno-oncology therapies such as immune checkpoint inhibitors. Now, historically, we have relied on immunohistochemistry tests assessing the expression of biomarkers such as PD-1 and PD-L1, which of course have been well validated both analytically and clinically and widely been used. But based on the observation that the PD-1 antibodies appear to work more often, though not always, in tumors known to have high mutational burden, such as lung cancer, both non-small cell and small cell, shown here and circled in red, and malignant melanoma. Many efforts are assessing the use of tumor mutational burden as surrogate biomarkers for immunotherapies. Next slide, please. So at Foundation Medicine and other companies as well, a lot of other companies where they offer genetic tests with large gene panels of 150 or more, they're looking at the correlation of patients with the high mutational burden and responses to tumor mutational burden. And this slide simply illustrates the impact of the tumor mutational burden in the different solid tumors. And as you can see, it's probably hard to read this slide. Malignancies at one end, such as esophageal carcinoma, small intestine adenocarcinoma, have very low tumor mutational burden, in contrast to malignancies at the other end of the scale, such as melanomas, squamous cell carcinoma of the skin, and of course basal cell carcinoma. They have a particularly high tumor mutational burden. Next slide, please. And if you look at the tumor mutational burden and survival analysis in patients with the malignant melanoma who were subjected to immune checkpoint therapy, work has been shown that the patients who have a high tumor mutational burden, shown in green in this particular curve, seem to have the optimal progression-free survival, in contrast to those with a low tumor mutational burden. Next slide, please. And this slide shows the relationship between overall survival and it's clear that the overall survival is also impact in the case of melanomas treated with this specific immune checkpoint based on the tumor mutational burdens. Next slide, please. But importantly, the jury is not in, because we're only just learning the biology behind the notion of using tumor mutational burden or other potential genomic surrogate markers. It's very clear that the tumor mutational burden is indeed a surrogate for the neoantigens, which are, if you like, proteins expressed on the cell surface, which are important for the immune checkpoints to interact with. But we're also learning that the tumor mutational burden and the immunohistochemistry results of the PD-L1 do not always co-associate. There have been instances where you have low TMB and high PD-L1, and you have a very good response, and vice versa. And lastly, but not least, the host genetics also seem to impact the response to immuno-oncology, as shown by about 20 to 30% of the patients who may have a high TMB and also a high PD-L1, but do not achieve a response to immune checkpoints. So I think there's a lot more work to be done here. But for the moment, the TMB should probably be utilized in the context of clinical trials, but the immunohistochemistry test can be utilized in the real world. Next slide, please. Let's talk a little bit now about the clinical outcomes or the clinical utility of the the information which is garnered by these tests and see where we are at the present time to see whether we can justify the expenses and, of course, sometimes the inconvenience of subjecting our patients to these tests. Next slide, please. Next slide. So here is a rather busy slide. I have collated a large number of high-quality clinical studies that have evaluated the use of personalized or precision cancer medicine. And this effort was led by a colleague of mine, Ian Tannock, based at Princess Margaret Hospital in Canada, and published in the New England Journal of Medicine about 3 years ago. And here, if you focus on the right-hand side column, which illustrates the principal outcomes of these particular studies, and most of these studies were randomized studies. They used different genetic tests, and the sample size in the selected studies were quite reasonable, ranging from about 250 in these studies shown on the bottom to just under 2,000 patients in the impact compact study. And what is very clear is when you look at the outcome at the present time, it seems that it leaves something to be desired. Most of the studies have shown that the benefit is being in very few patients. The response rate in selected studies may be as high as 20%, but the overall benefit in terms of survival, whether it's overall survival or progression-free survival, still remains to be less than 10%, and in some studies, it's even less than 5%. So I think at the present time, a lot of efforts are going on to embark upon more specific, well-designed genomic studies to see if we can determine the true benefit of utilizing precision medicine in the clinics. So next slide, please. So to exemplify how we are doing this, there are a number of new methodologies which have been introduced. And the first is known as the basket trial. And in the basket trial, we test different forms of cancers which contain the same mutation, which are then treated by the same drug. And for example, you can have a BRAF mutation in melanoma, or a BRAF mutation in a lung cancer, or a BRAF mutation in the colon. And we know that the functionality of the mutation may vary according to the tissue in which it is expressed. For example, the BRAF in the colon doesn't appear to be having the same functionality as the BRAF in melanoma. So this is being tested in basket trials further. So next slide, please. And another trial is the umbrella trial, where one kind of cancer is divided into subtypes based on the gene mutations, which are then targeted by a different drug. And these are also being tested. And to illustrate these examples, I will show you the next slide, please. One particular study, which is just opened up in the UK as well, is the complex study known as COPISCO. The CUP in the COPISCO stands for carcinoma of unknown primary. And this is a study which has 9 arms in the experimental arena. And the control arm, of course, is got the chemotherapy. And the 9 arms are very interesting because you have each arm is driven by a specific genetic mutation identified in these patients with carcinoma of unknown primary. And then the therapy is matched according to the mutation. And the last arm has immuno-oncology treatment as well. And the decision for the randomization are made after deliberation in a molecular tumor board. And this study started about 18 months ago. It's currently ongoing in 27 countries in the world, excluding the US and Canada. And the latest result will be presented at the ESMO meeting in September. Next slide, please. So we're coming to the end now. So let me finish off with some concluding thoughts. Next slide, please. So, and on these two concluding slides, I have put various things which I may not have had time to cover in full in today's brief lecture, but I've added them here because of the importance. So firstly, we should remind ourselves of the ability of cancer to evolve and adapt. And this is a principal challenge for treatment in general and for the paradigm of targeted therapies, including immunotherapy. And this perhaps is one of the main reasons why the current results are not as optimal as we anticipated. Secondly, the cancer adaptation, sometimes referred to as plasticity, is driven by intra and interclonal heterogeneity and contribute to resistance to therapy. And this also means that, if, for example, in the future, someone considers to do gene editing using, for example, the CRISPR methodology, that you have a mutation, why don't you use the genetic scissors and just simply clip it out? It probably won't work because of the extent of intra and interclonal heterogeneity. Additionally, some of the mutations can confer a selective survival advantage and increase proliferation following conventional or targeted treatment. For example, in patients with chronic myeloid leukemia, some of them respond very nicely to the tyrosine kinase inhibitors, but then they acquire a new mutation, which then creates problems, for example, in this case, T315I. And this can result in disease relapse, often due to the mutation resistance, mediated resistance. Next slide, please. And this is my last slide. The notion of functionality, and I've already addressed that when I was talking about the clinical trials, the functionality of the mutation based on the tissues. We also now recognize that different mutations can be present in the same areas of the tumor and the metastases, and the order in which the mutations arise can affect the phenotypic characteristics. For example, many tumors are driven by 3 different driver mutations. And if A comes before B, the phenotype may be less aggressive compared to if B comes before C, and so on. So this is something which also needs to be established. And last but not least, the availability of multiple techniques to test a patient's tumor is a challenge. And this also adds to the additional uncertainty. So as a final thing, much more work lies ahead. And my last slide, please. And I want to thank you all for listening, and I'll be happy to take some questions if you have.
So thank you very much, Tariq. That's been a very interesting presentation. And we're going to take a couple of questions from the audience. We don't have too much time, so we'll just go with 1 or 2. So if you've not already done so, please type your questions into the question box. And we'll start with the first question in the meantime. So Tariq, would be very interested to hear your answer of, is there a role for industry to incorporate companion diagnostic tests in new drug developments?
So I think this is critical actually, because in order to optimize and lower the cost of drug development, it's important, firstly, to identify as precisely as possible the target which appears to be more relevant than others in driving the pathogenesis of that disease. And then if a suitable, agreeable test is available to determine this test, this should be included in the development of that particular drug. And I think this particular concept is now been pioneered by a number of companies, and they're bringing in the companion molecular diagnostic test in the early phases, soon after the drug discovery, before the preclinical drug development takes place as well.
Thank you, Tariq, for answering. So we just have one more question that we're going to ask, and if anybody has other questions, please do just email us through to them and we can follow up after the webinar. So we wanted to ask as well, this is a question that's come up, are genetic tests useful when a patient relapses?
Yeah, I think so. In fact, I think in the real world, outside of clinical trial, this is probably the best example of utilizing a pan-cancer test when a patient has relapsed, because we may already have had a baseline, a simple genetic test for a targeted panel or a PCR done at diagnosis, which tells you that this particular driver mutation which is driving that cancer. And when the patient relapses, we do not really know what is the genomic landscape in that patient. So by doing a pan-cancer test, we will be able to detect the presence or absence of additional mutations and then assess if an approved therapy is already available for that tumor and then offer it to that patient. So I think it'll be fair to expand this and say every time a physician needs to make a clinical decision about the management of a patient, this should be a trigger to consider a genetic test, whether it's a tissue-based test or a liquid biopsy. Thank you.
Thank you, Tariq. So just a couple of words from us to finish up the webinar. We are the BIA, the Trade Association for Innovative Life Sciences in the UK. And if you're not a member and you're interested in joining, please get in contact or visit our website for more information. As you know, this has been episode 2 in our Bench to Bedside webinar series, and we hope you will all join us again for the next episodes on the 6th of September, 11th of October, and for our concluding panel session at the UK Bioscience Forum on the 17th of October. For more information to register for the next episodes of the Bioscience Forum, please visit our website www.bioindustry.org. Tariq, thank you again so much for joining us today for an excellent episode 2 in the Bench to Bedside series. And thank you also to everyone in the audience who could join us today. We look forward to reconnecting for the next episode on the 6th of September.
Thank you.